Hi guys, and welcome to my video. For this video, I'm going to show you how I made my very own 3D recyclable bacterial cell. For the materials, I used an illustration board, 2 folders. I also have stocked glaze, yarns, the combo scissors and glue, and my laptop for my preference. I started off by cutting the folders into pieces as the backbone of my cell, and then I covered it with a colored paper that I just found on my storage box. And here, as you can see, I grabbed my clay to start making the details for the structures of the cell. And here I added a green yarn to serve as my nucleoid. In this clip, I used my old art materials that I haven't used in a quite while. Attaching these yarns took me a while. Share ko lang. Now that you've seen how I made my diorama, I'm going to show you each parts of the cell. First up, we have the capsule. The capsule, which is made up of polysaccharides, keeps the bacterium from drying up and protecting it from this phagocytosis. Next is the cell wall, right here, the green one. The cell wall gives the cell its shape and surrounds the cytoplasmic membrane, protecting it from the environment. It is responsible for keeping the cell from bursting when there are large differences in osmotic pressure between the cytoplasm and the environment. Next, we have the plasma membrane, the thin layer over here. It encloses the interior of the bacterium, regulating the flow of materials in and out of the cell. And then we have the cytoplasm, the blue area, where functions for cell growth, metabolism, and replication are carried out. It is composed of water, enzymes, nutrients, wastes, and gases, and contains cell structures such as what is shown in the clip. The nucleoid, right here, is where DNA is located. It is not a membrane-bound structure, but it's an area where strands of DNA is found. And then we have the pili, these hair-like structures. These assist the bacteria in attaching to other cells and any surfaces. These red balls right over here are called ribosomes. These translate genetic codes from molecular language of nucleic acid to the building blocks of proteins. They are never bound to any organelles but are freestanding structures distributed throughout the cytoplasm. And this right here is the mesosome. It guides the distribution of duplicated bacterial chromosomes into the 2 daughter cells during cell division. They also carry the enzymes for aerobic respiration and increase the surface area for the same. These tiny clays over here are called inclusions, which are nuclear or cytoplasmic aggregates of stainable substances, usually proteins. They typically represent sites of viral multiplication in a bacterium or a eukaryotic cell, and usually consist of viral capsid proteins. And this right here is a plasmid. It contains genes that enhance the survival of an organism, either by killing other organisms or by defending the host cell by producing toxins. Some plasmids facilitate the process of replication in bacteria. And lastly, the flagellum, which primarily is a mobility organelle that enables movement and chemotaxis. And that is it. I hope you guys gained something from my video. Bye!